So today we will discuss about the type 2 hypersensitivity reactions, which is also called as the cytotoxic reactions. So we'll further go. So imagine that, so firstly I'll draw a red blood cell, which is a biconcave disc-like structure. So imagine this is a red blood cell and the person takes some kind of drugs. In this case we'll have penicillin. So some part of the drug tends to bind to the surface of the red blood cells. So what happens here is, previously the red blood cell was our own cell, alright, OWN. So this was our own self cell, and as soon as some exogenous chemicals started to bind on the red blood cell surface, now this cell is assumed to be a foreign cell, so a non-self cell. So whenever there is some foreign objects or chemical substances, our immune system gets activated. So as soon as this penicillin is bound in this red blood cell, the antigen presenting cells come and try to opsonize this part of the drug and tend to present it on its surface. I've done already all this process in the type 1 hypersensitivity reactions. So if you have any doubts, you can go to the previous video and check the first two stages. So this is a MHC2 molecules where the antigen presenting cells like dendritic cells. So this is a dendritic cell, so which has opsonized a part of the foreign antigen and presented it as MHC2 cells. And the T helper cells come recognizes because they have a specific receptor for this MHC. So imagine this is a T helper cell, and this T helper cells sends information to our B cells. So write B here. Our B cells that there is a foreign intruder, and the B cell starts to proliferate and divide into plasma cells, which is a subset of the B cell. And the plasma cell starts to secrete antibodies, and all foreign chemicals, we have specific antibodies to them, and whenever there is a chemical change in the structure, the IgG and IgM are produced because they have high affinity towards the exogenous chemicals. So there is production of IgG and M. So IgG and IgM are produced, and as soon as these antibodies are produced, these specific antibodies, in this case we take it as IgG, starts to go to the site of the action and try to kill this foreign antigen, which is now a part of a cell. So this is the problem. The own antibodies thinks that the self cell is some foreign object and tries to kill the own cell. So there are three important mechanisms in the ways that these antibodies kill the own cells. We'll see what are these three mechanisms. So the first mechanism is through phagocytosis. So in the phagocytosis, when the erythrocytes or red blood cells structure is changed, it would automatically become the target for our phagocytic cells. So these phagocytes specifically go down. So as soon as the immunoglobulins G and M are formed, the immunoglobulins G come to the site of the action and bind to the external antigen, something like this. And as soon as the immunoglobulin G has started binding towards the antigen, there is release of complement proteins, C3 proteins, which include C3A, C3B and C3C. And as soon as the antibody has started to bind, it also recruits other phagocytic cells like macrophage cells and dendritic cells, alright, M for macrophagic cells and dendritic cells, to come to the site of action and undergo the process of phagocytosis. So the dendritic cells and the macrophagic cells try to phagocytose the red blood cell membrane and ingest the opsonized cell. So this is the first mechanism where there is cytotoxicity through the process of phagocytosis, and we'll write the important points. So in the phagocytosis, due to complement proteins, C3 complement proteins, and there is also high affinity for the Fc tail of IgG antibody, so that all these phagocytic cells come to the site of the action, and due to high affinity towards the Fc portion of the antibody, that is the IgG in our case, so the macrophagic cells come to the site of the action and there is process of phagocytosis. And now we'll talk about the second mechanism of how the cytotoxic reactions happen to the cells, and this is through the inflammation, that is the complement mediated cytotoxicity. The main player here is the complement proteins. So complement proteins. So how is the second mechanism? We'll go back to the picture, and here we have the red blood cells which is already bound by the antibody. This is IgE antibody, IgE. So once the antibody is bound to the antigen, there is activation of the complement system through a classical pathway. So there is 3 different pathways to activate the complement system, and one of the pathways is the classical pathway, where there is release of several complement proteins, and one of the main function of this complement protein is they tend to recruit neutrophils and monocyte cells and cause inflammation in the body. So there is recruitment of the neutrophil cells and monocyte cells, and these cells tend to cause inflammation in our body. So this is how the second mechanism, that is through the complement proteins. And now we'll see about the third mechanism, how the cell is. So here there is recruitment of the neutrophil cells and monocyte cells. So these cells cause inflammation reactions. In that way the cell acquires toxicity. And there is another third type where there is antibody dependent cell mediated cytotoxicity. So we'll just simply write antibody mediated. So in this third mechanism, the cells like macrophages, neutrophils and natural killer cells, that is macrophages, neutrophils and natural killer cells, that is NK cells, come to the site of action, and all these 3 kind of cells have specific enzymes and proteins which they spill on the cell. So they tend to spill all their enzymes and proteins, which involves in destruction of the red blood cells. So there is no process of phagocytosis, but through the proteins like perforins and granuzymes, they kill the cell. So this is the third mechanism of how the cell undergoes cytotoxicity. And there is another type of reaction. This is called as the antibody mediated cellular dysfunction. So before that we'll write the type of the cells which are involved in the antibody mediated cell mediated cytotoxicity. So we'll write NK cells, that is the natural killer cells, macrophages and neutrophils. They cause the lysis of the cell through enzymes. So there is no process of phagocytosis, just through the enzymes they tend to kill the cells. There is another reaction that is very specific, and this is antibody mediated cellular dysfunction. So we'll read antibody mediated cellular dysfunction. So this also undergoes similar process, but here the antibody does not cause harm to the cell but changes the function of the cell. So there's no harm to the cell, but the function of the cell is altered because the antibody is now affecting the receptor zones of the particular cells. Example, we have a classic example in Graves' disease and Myasthenia gravis. So firstly we'll see Myasthenia gravis. So this is a condition where the antibody tends to change the process of the particular cells. So firstly we have Myasthenia gravis. So in the Myasthenia gravis, so we have, this is the axon cell and this is the muscle, and here are the acetylcholine, and these are specific receptors for the acetylcholine. And what happens in this case is the antibodies tend to bind on the surface of the acetylcholine receptors. So in this place there is binding of the antibodies, so there is no place for the acetylcholine to pass information to the muscle cell involved in any kind of neurotransmitters between the axon cell and the muscle cell. So eventually the muscle cell gets weak because there is no neurotransmission signals between the axon and the muscle cell, because the antibody is now placed in the acetylcholine receptor. So this is what happens in the Myasthenia gravis. So the antibodies which is now bound doesn't cause any harm to the muscle cell, but there is no neuromuscular transmission between the axons and the muscle cell, which makes the muscle cell eventually weaken. So this happens in Myasthenia gravis, and there is another classic example for this type of antibody mediated cellular dysfunction, and the other type is, the classic example is the Graves' disease. So we'll write, so Myasthenia gravis, where the antibody binds on acetylcholine receptor place and blocks the neurotransmission. The neuromuscular transmission, which leads to weakness of the muscle. And another example for this kind of cellular dysfunction is Graves' disease. So what happens in the Graves' disease is, so in the Graves' disease, for example, imagine this as a thyroid cell. So this is a thyroid epithelial cell, and it bears receptor for the thyroid stimulating hormone. So the thyroid stimulating hormone, so imagine this is the receptor for thyroid stimulating hormone, has to bind to the epithelial cell of the thyroid cell in order to say that you have to produce certain amount of thyroid hormones. But what happens is, in this case there is binding of the antibody instead of the thyroid stimulating hormone. So what happens is, the antibody is now bound to the thyroid cell. So the antibody stimulates the thyroid cells to secrete more amount of thyroid hormones, that is T3 and T4. So what happens is, even though our body has adequate amount of thyroid hormones, the antibody which is now bound to the thyroid epithelial cell of the thyroid is now asking the thyroid cell to produce more and more amount of thyroid hormones, which eventually leads to hyperthyroidism. So this is what happens in, so I'll write hyperthyroidism, I'll write hyper T. So this is what happens in the Graves' disease. So the antibody eventually doesn't cause any harm to the thyroid cell, but instead it stimulates the thyroid cell to secrete more amount of thyroid hormones, which will result in hyperthyroidism. So these two examples, Myasthenia gravis and the Graves' disease, The antibodies